welcome everybody back, both on Zoom and in person. And for the second session, this is going to be moderated by our own Roger Peterson. So I will hand over the podium to him.
Thank you very much, Virginia, and everybody both in the room and on Zoom for your attention and participation in the symposium. Our next speaker being Dr. Winn. I'm very happy to be able to introduce you as Associate Professor in the Department of Obstetrics and Gynecology, Director of the Reproductive Stem Cell and Perinatal Biology Division, in which I and the other basic science members of the department inhabit. Virginia received her medical degree as well as a doctorate in biochemistry from the University of Rochester and completed residency and fellowship in maternal-fetal medicine at UCSF. Dr. Winn's research program, as all of you I'm sure are well aware, focuses on the development of the placenta, particularly the complications of placental development inherent in preeclampsia. Among her many impressive achievements, she has 2 NIH studies. I won't go into too much detail and take all of your time, but needless to say, is an inspiration to all of us within the department, and we're very much looking forward to your talk.
All right, thank you very much. So what I'm going to be talking about today is the teaser was, what do fat and sugar have to do with preeclampsia? I took a little liberty, and fat is really going to be referring to leptin, which fat makes, and sugar is referring to a molecule that recognizes sugars at the maternal-fetal interface. So preeclampsia is a condition that is very near and dear to my heart and well known by any obstetrician. It affects roughly 3 to 8% of all pregnancies, and it's characterized by a new onset of hypertension and proteinuria or other end organ damage after 20 weeks of gestation. It has a strong clinical impact worldwide, accounting for 70% of maternal deaths. In the US, it accounts for 18% of maternal deaths and 40% of fetal mortality. The only definitive therapy is delivery of the placenta. And in the US, due to the fact that you need to move towards delivery, accounts for about 20% of preterm births. So a major indication for preterm birth. The placenta, my favorite organ, is the known etiologic organ for preeclampsia. Here I'm depicting a cross-section of the human placenta. Do I have the mic? From a 3rd trimester placenta. And what you can see is that the placenta is composed of a tree-like structure that is growing from the surface of the placenta that is facing the baby in the amniotic fluid. And the placenta is attached to the maternal uterus via anchoring villi attaching to the maternal decidua, which is the hormonally modified lining of the maternal uterus. And my lab has focused a great deal of effort on understanding this decidual basalis or basal plate region of the placenta, because that is where the fetal trophoblast cells invade into the maternal tissue, are involved in remodeling the maternal vasculature, anchoring the placenta, and also likely a very important site for development of maternal fetal tolerance. So this basal plate region is where the fetal cells from the placenta of that chorionic villus tree structure anchors into the maternal decidua, and fetal trophoblast cells actually migrate from the tips of the anchoring villi into the maternal tissue and remodel the maternal vasculature. So this region of the placenta is really a chimeric portion of this organ, a combination of both a maternal genotype and a fetal genotype cells interacting without rejection, which is a remarkable immunologic aspect of human placentation. So how we think about preeclampsia pathogenesis traditionally is that it is a two-step process, that while it presents in the latter half of pregnancy, starts to develop with poor placentation in the first half of pregnancy. And so a poor placentation is reflected by poor remodeling of the maternal vasculature. So you get poor perfusion into that intervillous space. And that leads to oxidative stress of the placenta, releasing a number of factors, toxic factors. You know, the old term of preeclampsia is toxemia, because we think about the placenta putting these toxic factors out into the maternal circulation to make the mom sick. But these factors are known to cause endothelial dysfunction of the maternal vasculature, as well as an enhanced inflammatory response. And these initially were thought to be two independent rival concepts, but they're really interdisciplinary in terms of interaction. And a lot of this really explains the clinical signs and symptoms we see in preeclampsia. So early in my postdoc, I was interested in understanding that basal plate region of the placenta, and so collected that region from both normotensive pregnancies and preeclamptic pregnancies, and performed gene expression analyses. And as you can see here, the very top gene that came out that distinguished the preeclamptic placentas from the normotensive was leptin. The association of leptin with preeclampsia was not new at this point. So this was reassuring that we saw this hit, but this was actually the first time it had been shown in the basal plate region of the human placenta. And so thinking about leptin and preeclampsia, we know that leptin levels actually go up in pregnancy compared to outside of pregnancy, as well as in preeclampsia, they were at much markedly elevated levels. And what was known also was that the serum levels of leptin were elevated even earlier in pregnancy before the presentation of the clinical syndrome. And so even by 13 weeks gestation, there was an increased risk, if your leptin was elevated, of developing preeclampsia later in pregnancy. There was also this association, that known clinical association, that women with high BMIs or obesity are at higher risk of developing preeclampsia, likely a connection with this hyperleptinemia, because leptin levels are influenced by the amount of adiposity one has. And there had also been a few studies that had found gene polymorphisms within the leptin gene associated with preeclampsia. And actually, what I want to show here is that in a number of much larger studies, both of these are from groups at Stanford that had looked at proteome and metabolome in an unbiased way in cohorts of women with developed preeclampsia or normotensive. And what you see here is on the left, which is a proteomic analysis, that actually leptin, it really distinguishes the difference between preeclamptic and normotensive, again, before 20 weeks, even before the clinical symptoms present. and is markedly elevated across the whole gestation, where some of the other well-known SFLT1, other factors that we hear associated with preeclampsia, you can see are only showing distinctions later in pregnancy. So leptin is a pretty profound phenotype here when you're looking at the longitudinal differences. And this is at the, from metabolome work, again, looking at first, second, and third trimester. And again, you can see of all of the genes that they did show that were distinctively different in some common players, leptin really helps really distinguish the cohorts in the population. So as a placental biologist, I was interested in thinking about why is high leptin levels leading to this clinical presentation. And the underlying hypothesis of preeclampsia for the majority is that we have this poor placentation, which is a reflection of poor trophoblast invasion and vascular remodeling. So a graduate student in my lab was interested in looking at the effect of leptin on trophoblast biology, specifically trophoblast invasion. And what she showed was that actually, with increasing levels of leptin, again, 10 is kind of reflective of normotensive or the lower end of pregnancy levels. 80 is more reflective of normal pregnancy in the third trimester, or in normal pregnancy, and 160 and 320 are clearly preeclamptic levels. Those are not normal levels. And what you see is that the degree of trophoblast invasion increased with leptin, but at the very high levels, that enhanced invasion that you see was abrogated. And so we found this very interesting in terms of understanding the leptin biology. She confirmed that this was not due to any toxic effect or proliferative effect on the trophoblast cells themselves in response to leptin. And then she confirmed this data. The HTR8s are an in vitro trophoblast cell model. She isolated primary cytotrophoblasts from early placentas and showed a similar pattern of this, what we call a biphasic response of trophoblast invasion to leptin. So she showed that moderate levels seem to promote invasion, but high levels, you no longer see that promotion, and although not statistically significant, maybe even inhibition, but it's more a lack of the invasion promotion. So she considered the leptin receptor. There's different isoforms of the leptin receptor, short and long forms that can lead to different signaling pathways. She did a lot of work looking at these different pathways, and what I'll show you is the results that she determined that trophoblast invasion is ERK dependent, and she showed this by using inhibitors of the ERK pathway. And what you can see is that you see phosphorylation in response to leptin of ERK that is inhibited in the presence of the inhibitor. And then when you look at the invasion piece, as we'd seen previously with the control, we see this biphasic response. In the presence of the inhibitor, you lose the biphasic response. You don't inhibit beyond the basal level, but you inhibit the enhanced invasion. And this is just showing that this was not due to changes in cell number in response to leptin. So summary of this leptin was that leptin appears to have a sweet spot in terms of maximal invasion of trophoblasts in around the 80 nanogram per mil, that this is ERK phosphorylation dependent. But all of the signaling data that we've done didn't help explain why we're seeing this biphasic response to leptin. So what I want to point out is if we go back to the microarray work, there was another molecule that was significantly upregulated in preeclampsia, and that is Siglec-6, which is a sialic acid binding immunoglobulin like lectin. And it was recently at this time shown that Siglec-6 actually was cloned based on the fact that it binds leptin. So here we had leptin elevated in preeclampsia, Siglec-6, a receptor for leptin, a non-canonical receptor for leptin. And at that point, this was one of the first reports of this association of Siglec-6 with preeclampsia. And this has been confirmed multiple times since that time. So the Siglec-6 is a part of a much larger family of Siglecs. These are transmembrane receptors that bind sialic acid sugars on other molecules. There is part of the family is highly conserved across many species, but there is also a rapidly evolving family that is thought to be evolving due to the need of the immune system to recognize and pathogens and our evolution, rapid evolution with our surrounding microbiome. The signaling capacity, this is a depiction of Siglec-6. It has the recognition domain for the carbohydrate, transmembrane domain, and then it had two potential signaling motifs. And we have gone on and showed that those tyrosines in these motifs are phosphorylated, that is a Src kinase dependent, and that both need to be phosphorylated to recruit SHP2 to this activation site for downstream signaling. So this is just showing that Siglec-6 is one of many members of this family of receptors. And what you can see is that most of the cells that express these are in our immune system. And each immune cell tends to have its flavor of Siglec-6 or its pattern of Siglec-6. The trophoblast is actually, Siglec-6 is specifically expressed on trophoblast. So again, kind of thinking about the immune interaction and this foreign tissue and invasion into the maternal decidua, this restricted expression pattern definitely caught our eye. So we were interested in following up on this Siglec-6 expression in trophoblasts. This is RNA analyses of normotensive pregnancies across increasing gestation compared to preeclamptic pregnancies across a similar gestational age window. And you can see Siglec-6 at the RNA level is markedly elevated. Also, you'll notice that it appears that Siglec-6 increases at much lower levels, but has a gestational age dependence with its expression as well. This was confirmed also by immunoblot analysis at the protein level. Again, we see elevated levels of Siglec-6 in the basal plate region, which was our initial interest, but also in the chorionic villi. That's those floating villi that are bathed in maternal blood, also have increased levels of Siglec-6 in preeclampsia. This is the immunohistochemistry showing that the cell type that is expressing Siglec-6 is indeed the trophoblasts syncytium that are lining the chorionic villi, as well as invasive cytotrophoblasts within the maternal decidua. So this is just showing increasing gestational age of 24, 30, and 36-week placentas. CK7 shows you what are the trophoblasts in the tissue sections, and then the middle panels are the Siglec-6 expression, both in normotensive and preeclamptic. So the next question was, is Siglec-6 interacting with leptin in a manner that could be explaining this trophoblast invasion? And so Kristy in the lab took the HTR8 cells, again, now overexpressed Siglec-6 in them, and treated them with increasing leptin levels. So this is just showing that Siglec-6 decreases the overall number of cells, but that there is no leptin dependence in terms of number of cells, which is important when you start to think about the invasive data. So as we'd previously seen, you see this biphasic response to leptin for trophoblast invasion. In the presence of Siglec-6, we're seeing a similar pattern. So there wasn't a big change in trophoblast invasion in the presence of Siglec-6. In parallel experiments, we also utilize a different trophoblast cell line in the lab, the BWO cell lines, which is a choriocarcinoma cell line. And we also looked at invasion with that cell line and found a completely different response. So again, this is the BWO cell lines with empty vector had this biphasic response. In contrast, with no leptin around, overexpression of Siglec-6 had significant increase in invasion, 6 to 8-fold, and that is by far the most invasion we'd seen in any manipulation of trophoblast cells. And you can see that with increasing leptin, you inhibited that invasion. So clearly, the cell context is very dependent, very important. So the HTR8s versus the BWOs gave us totally different responses. And one explanation may be that the choriocarcinoma cell lines have very different expression of sialotransferases. And remember, Siglec-6 binds sialic acid carbohydrates. So that may help explain the difference. But what this showed to me was that it's really important what your in vitro model system is that you're using. And so I was very happy that in 2018, it's kind of the year of the placenta, there was a bunch of really key and important papers in human placental biology that came out at this time. One was after 30 years of knowing how trophoblasts are, you can generate human murine trophoblast stem cells, hadn't been able to figure it out in the humans. Kind of a common theme for the morning session of can't always translate murine to human when it comes to understanding development. But this group in Japan figured this out. And so we have in our own lab now developed these Okaya human trophoblast stem cell lines. This is Shilpi Segal's work, a postdoc in my lab. She's taken these human trophoblast stem cells and generates spheroids, showing the CK7, which is trophoblast marker, E-cadherin, which is a cytotrophoblast marker, and GATA3, another transcription factor expressed by trophoblasts. And she's stained these and showed that these trophoblast stem cells express Siglec-6. So we are going to be now utilizing this model system. She's in the process of generating knockdowns, Siglec-6 knockdowns. So we'll have Siglec-6 expressing knockdowns, and then there's a bunch of different mutants of that Siglec-6 molecule, both for signaling and ligand binding that we will be working with to understand this biology. So kind of the current focus in the lab is we'd like to determine factors that regulate Siglec-6 in expression. What I didn't mention was we know across gestation that Siglec-6 is on early, it's off, and then it comes back on again in a normal placental biology. We wanted to what are the ligands of Siglec-6 at the maternal-fetal interface? Those are not known. define downstream impact of Siglec signaling, and begin to understand what is the interplay between leptin, the leptin canonical receptors, and Siglec-6 in understanding trophoblast biology. So with that, I'd like to thank the funding sources I've had. This is a current picture of the Winn lab, and I'm happy to take any questions that you might have. Kristy.
Hi, Virginia. That was excellent. Thank you so much. I'm curious because that was the year of the placenta, and if I remember correctly, some single cell
Yes.
sequencing data came out. So I
Yep.
was wondering how similar the induced trophoblast cells from your protocol are to the actual trophoblasts in vivo.
Yeah. So there are a number of papers that have even Okaya utilized RNA-seq to show blastocyst, early human placentas and their cell lines, and they appear to be a robust model of trophoblasts, human trophoblasts. Yes.
Play into postpartum preeclampsia?
Yes. Yeah. Yes, I'd be happy to thank you. So the question is basically, how do you think about postpartum preeclampsia when we think about preeclampsia being driven by the placenta? And how, once you deliver the placenta, why do these women present with a preeclampsia picture at that point? And yes, a lot of hand waving, but I think my current thought process, which is nothing to do with this data, is that part of the endothelial dysfunction is happening because of balances of different factors that the placenta is making. And it may be that as those factors are being cleared by the kidney and their half-lives, that things that were kind of on the border generate that imbalance that presents as preeclampsia. That's my best hand waving, and I would love to actually test that hypothesis.
I don't know if there's any questions from Zoom, but we're just one more question, then we'll have to move on.
I have kind of a clinically relevant question, which is, since not all obese patients develop pre-E,
Yeah.
if you control for obesity and specifically central adiposity, is the leptin, the very high leptin, still predictive of preeclampsia development?
Yeah. So in the paper that looked at the association between leptin levels and preeclampsia, it still held true even controlling for BMI, and that is because the placenta actually produces leptin. So the leptin that is circulating is a combination of maternal leptin from fat sources, but also from the placenta. So it's probably a reflection of the stressed placenta, is how I would think about that. Great.
Okay, let's give Virginia another hand. We'll move on to our next speaker, who is Michael. Michael Angelo is a board-certified pathologist and assistant professor of pathology in Stanford School of Medicine. He received his doctorate degrees from Duke University and completed his residency in clinical pathology at UCSF. He's a leader in the high-dimensional imaging and expertise in tissue homeostasis, particularly pioneered multiplex ion beam imaging by time of flight, or maybe TOF, which uses mass spectrometry to identify a multitude of epitopes in a single specimen. They're quite a revolutionary technology. Very much looking forward to hearing you. And nice to see you in person.
Yeah, you too. I was going to say.
Meeting,
Good to see you.
I'll put my hand up when you've got 5 minutes.
5 minutes? Okay.
I planned for like 15-ish. Is that right? Okay. That's right. Hey, really happy to, oh man, I'm really loud, aren't I? I'll put it further down. Is that better? Not so? Okay. Really glad to be here. I'll just jump right in and talk about some of the stuff we've been doing. This work will, I think, hopefully be out and published next month after a long and arduous review process. We had 7 reviewers over 520 days. It was extraordinary, but I think it's over now, so that's good. So hopefully that'll be out next month, and we're really excited to talk about some of the work that we did in that paper and what we think we're going to do next. Trying to leverage, archival historic sources of tissue from human pregnancy to get kind of a window into some of these hard to observe human specific facets. So in general, my lab, we're really interested in sort of understanding how tissues in human bodies are sort of, the whole is more than the sum of its parts. And so if we think about, sort of this nested organization that, mammalian systems have, so you have cells that can take on a multitude of different functional states, and then those can be put together into multicellular units like a gland that have some sort of composite function that all the cells carry out together. But all the while, right, that's kind of interconnected with the functional state that those single cells have as well. So it's kind of this interdependent dynamic that leads to the composite response that we observe, like in tissues and whole animals. And so the way that we think about this is from the level of trying to understand tissues in terms of reoccurring themes in organization, and we call that sort of collectively a spatial ontology. And so, so just giving you an idea of the specifics, what that means, it's sort of, we can think about tissues being comprised of the different cell types and how many of them there are different types of tissue. And then we can also think about it in terms of sort of like you can see in the middle, what's the conserved blueprint or structural plan? So, do they have sort of concentric organization of tissue layers, sort of in, onion skin kind of fashion? And then lastly, we can actually think about it in terms of what different cell types are when they're close to one another. What kind of relationships do we see? So are there some cells that occur closer to one cell type than you'd expect them to by chance? And so we can tally all those things up and think about it in terms of like a set of rules. And then we can compare across lots of tissues. And a lot of times we can find not only like shared sort of, plans for how these tissues are put together, but you can a lot of times see relationships linking to diseases that seem really unrelated. And by doing that, what we can do is we can start to kind of look at those shared ontologies, and we start to infer functional mechanisms by pulling in information from fields that are oftentimes pretty siloed, and as well as think about how they could be used for clinical assays and drug repurposing. And so the platform that we're using now in the lab is kind of most of the studies utilize archival formal and fixed paraffin embedded tissues. We try to maintain compatibility with those specimens so we can tap into historic, without having to prospectively get frozen material. We have 3 main spatial platforms that we're using right now to look at sort of spatial whole transcriptome, spatial proteomics. I'll show you that one in a second. And that's what we've done the majority of our previous work with. And then we're now have de novo mass spec imaging in-house that we're using as sort of for mapping hundreds of glycans at one time. And so, so just to kind of focus on this one for a second. So maybe it's pretty much, if you understand immunofluorescence, you'll understand this. It's pretty much the same thing. It's just more channels. And so we take a tissue section and we stain it with a cocktail of antibodies. And instead of them being tagged with fluorophore or something that'll emit light, they're tagged with a reporter that has a unique mass. And so then instead of reading out a protein based on it emitting, a specific color like red, we're reading it out by saying it has a specific molecular weight, like 142, right? So once the tissues are stained with those antibodies, we put it in a mass spec, and we go pixel by pixel across the area that we want to image. And an ion beam is focused down in a really small spot, and it has massive +1 charge xenon ions that hit the sample and liberate those tags as secondary elemental reporters, and we take those to mass spec and measure them. And then, long story short, we can, put Humpty Dumpty back together and kind of get something that looks like immunofluorescence. And so this is just kind of like an example of some images from different studies. About half the stuff we do is looking at cancer. The other half is looking at things like, I'm going to talk about today, spiral artery modeling in the maternal-fetal interface. Have a lot of work in collaboration with the Bindle Lab, also looking at sort of neurocognitive decline. And so right now we generate somewhere in the order of about, probably 50 images per day with these systems upwards. And usually it's about 40 plex protein imaging, and that ends up being about 100,000 cells per day. And so just to give you an idea, this is one cohort that we just recently acquired. That's the TNBC tonic trial that's looking at how patients with metastatic relapse in TNBC, how they do with nivolumab therapy. And so this is the kind of the cohort design. And like, here's an example of like, kind of a mosaic of just 3 channels of about a third of the data set. So the main thing from that is just kind of to give a qualitative estimate that it's just, you can't look at it manually anymore, unfortunately. So the last 2 or 3 years we spent putting together a pipeline to be able to process these data sets sort of at scale in a way that we felt like, you could actually trust what we were getting out. And so part of that was like things that seem really straightforward, but aren't, like just thresholding the images. You think that'd be really easy, establishing like a cut point for positive or negative, but that ended up being a more complicated problem than we thought it would. But we think we have something pretty automated for that can be done without a lot of human intervention. And then once we've done that, we developed this deep learning pipeline in collaboration with the Van Balen lab that can draw borders around cells with about the same accuracy as a manual human annotator. And you can see that here, that essentially, if you compare the results, the machine results versus the human results, they're pretty indistinguishable. And we sort of validated that across 15 different human tissue types. So that automated that part, which had also taken a very long time to do. And so then we can kind of get to the fun stuff, which is looking at sort of dimension reducing these high-plex data sets, so that we can see like what types of cells are there. We can do that with cell clustering. And then we can also look at things at the pixel level, because a lot of times there's features that don't necessarily respect cell borders that are interesting that you want to know about, like maybe collagen or extracellular matrix, or in the brain, that's a much bigger deal. And so then once we have that, we can start thinking about some of these features that we just talked about. So this is sort of, once we have everything done, this is kind of one of our main sort of graphical representations we use to look at an image. So this is what we call a cell phenotype map. So essentially all the cells have been outlined from the original image. And now what we've done is we've taken away all of the original protein imaging data. And then at each one of these, cell circles, we've just assigned it a specific color for what type of cell it is. So you don't have to worry about the color mixing. So, the color mixing, like how RGB overlay, that no longer matters. So these are categorically colored. And so you can see here, there's probably about, in this image, I don't know, something like 10, 12 cell types or something like that. And so that gives you an idea of the spatial distribution on a cell type basis. But then we can take that and we can look for, are there groups of cell types that tend to occur together one another? And it can even be like a certain cell type likes to hang out with itself, right? So you could kind of say that maybe about that lime green population in the upper left-hand corner. And so here is what we call like a neighborhood map, which is instead of the cells being colored by what types of cell they are, they're colored by what neighborhood they're in. And then for each of these neighborhoods, we have essentially like a profile to say who usually lives in that neighborhood. So it's like hipsters live next to coffee shops kind of thing, right? So, so now kind of getting to the stuff I'm going to talk to you about today. There's a lot of parallels actually between cancer progression and pregnancy, and I've been really fascinated by this. And Virginia gave an excellent sort of, preview for what we're going to talk about now, which is, sort of the role of these cells at the maternal-fetal interface, particularly trophoblasts. And so, there's if you look at sort of the steps of placentation, have like as she was saying, it's a pretty remarkable thing that that the placenta isn't just rejected, and so there's this very refined, elegant process for sort of carving out this hole in maternal immunity to allow it to stay there, all while not, succumbing to infectious disease and all that during pregnancy. And the other thing that goes along with that, right, is a placenta is actually pretty invasive, but it's not too invasive, so most of the time it doesn't go too far, it comes in just right. So I think there's a lot that we can learn from sort of watching the temporal progression of this, and particularly for this sort of differentiation trajectory from the anchoring villus. And so, we've got this trajectory from sort of villicide trophoblasts to extravillous trophoblasts, which invade into the decidua. These cells are, it's kind of remarkable because if it's sort of like if you think that a cancer cell can do it, it's almost everything I've played this game. Like I'll look and you'll look at different pathways for different sort of cancers and malignancies, and then you'd be like, oh, I wonder if every single they do all of it, right? So they promote ECM remodeling, recruitment of macrophages, immune suppressive properties. They have tolerogenic interactions with T cells, and then there's also all these other sorts of things. Like for instance, there's antigen specific, fetal specific regulatory T cells are induced during pregnancy. We don't necessarily know how at this point, but we know that's now a big gateway in order to get metastasis after lymph node involvement with cancer. And the other thing, I just saw a paper the other day I wasn't aware of, but apparently, they're now seeing that, women after their first pregnancy you can see there's actually evidence of CD8 T cell exhaustion in women, much like, was sort of the big breakthrough in CTLA-4 therapy in the early 2010s for looking at melanoma and renal cell carcinoma. So, there's all these things where, how it all exactly fits together is not exactly clear, but there's a lot of things that kind of align in a very interesting way. And so we were really interested in this process of spiral artery remodeling, which is that because, again, it's just absolutely wacky that this even happens, right? It's like trophoblasts, like invade the decidua, right? They're just going through there, that's fine. Nobody says anything, and then they actually get up to the blood vessels. And then during the first 20 weeks, these blood vessels lose their smooth muscle. The trophoblasts get into the artery and they actually replace part of the endothelium inside the artery. And this is totally fine. And it happens. And then it's baffling that, this even happens. And so this is sort of, you start off with a small condensed artery on the left. And by the end, you get something with the remodeled artery. It kind of looks like what you have on the right, where it's very dilated. And so you can accommodate the amount of blood flow that needs to come to the placenta during pregnancy, but you get to keep the velocity very low. So there's not near as much shear stress. And that from what I've read from people that have studied this a long time, they think is the adaptive role of having this sort of large degree of vascular remodeling. And as Virginia alluded to, this is known to be perturbed in preeclampsia. So that's kind of, another hope for why this is a really important process. These arteries are under remodeled. And they're still remodeled some, but it's clearly impaired, and the vessels seem to be smaller than they should be. So, and for the sakes of this study, we were mainly interested in understanding, how it happens in normative pregnancy. So, our main goals were sort of first understand like how the deciduous structure and composition sort of changes in the first 20 weeks, and then the second goal was to understand, kind of use this as a test bed to understand which of those changes were sort of driven temporally just by gestational age versus which ones looked like they were under more of local regulatory influences around the arteries. And so these are the three people that led this study, and they Shirley, Aaron, and Inna. And they put together this tissue microarray of 66 patients that underwent elective termination at SF General. And these were all for non-medical reasons. And we went through all these tissue blocks and found regions that had spiral arteries in them, and those were recorded and put into the tissue microarrays. And so we stained it with this large panel of antibodies with the BIVI on it. And then the other serial section we actually did, we were able to target some of these same regions of interest that we did BIVI on in a serial section, and did transcriptomic profiling on that with NanoString DSP. And so, what I'm going to show you now is just kind of some results from that study, and you can kind of think of all of these as being trends that we found that seem to be driven more by gestational age, just had a very strong temporal component, irrespective of the status of spiral artery modeling and what was going on with the tissue structure, versus those that looked like they were tightly, influenced by what was going on locally, like in that image. So they didn't look like they had but at least they had some degree of local modulation beyond that systemic component. So time is easy. We get that variable for free from the EMR, so we don't have to calculate that one. But space and sort of morphologic trajectories are a little tougher. So the first thing we did was we had arteries at each one of these stages of remodeling. So they start off really small, and then by stage 2, you can see they're getting bigger, and it looks like the smooth muscle, which is that pink layer around them, has been disrupted and it's fragmented. By stage 3, you've lost a lot of that. By stage 4, the trophoblasts surrounding the arteries have actually invaded in. And then by stage 5, you can see that they've replaced some of the maternal endothelium. So the first thing that was very interesting was you could see that this process, it is correlated with gestational age, but there's definitely, variability. And so, you could see even at week 20, there's still some arteries that are at stage 2, and you see arteries that are all the way at stage 5 as early as like week 14. So, there seems to be definitely some degree of variability within the decidua basalis. So the next thing we did was we essentially went and calculated a single 5 minutes. Single score, a single number to be able to rate where along this, remodeling trajectory we were. And once we had that one number, we could then, in the matching transcriptomic data for each of those images that we looked at with MIDI, we could look to see which transcriptional programs were correlated with spiral artery remodeling. And so then we came up with sort of in the paper, we've got the sort of a global readout of the transcriptional programs that are changing and going up and down in this process. And so you see some that are, going down the whole time, going up the whole time, but then you also see an interesting bunch that goes up and then comes down. So there's several multimodal patterns there. Just based on the immune cell composition in the decidua alone, we could predict what the gestational age was within like 15 days. And the thing that would seem to be really, really tightly correlated was the macrophage to the maternal NK ratio. And then lastly, what I was going to show you is just sort of some cool stuff from the targeted transcriptomic profiling of these trophoblasts, depending on where they were. So we compared trophoblasts at the cell column, the ones that were sort of immediately around the blood vessel, which we call perivascular, as well as these that were intravascular and inside the artery. And ultimately what we found was we had these, again, we saw these interesting upregulation of these transcriptional programs that are mostly associated either with cell motility or immune invasion. And many times the immune evasive pathways, again, were ones that have been associated with anti-phagocytic signals that you see in cancer, like with CD24, as well as C5-ORF30, which is one that's now been, I think, renamed MACIR, M-A-C-I-R. To come full circle to somewhat Virginia said, since we had the sequences of these EVTs within the arteries, and we actually had the transcriptomic information for the arteries around them, we could use this program called NicheNet to infer what the cell-cell interactions were that were going on between the different cell types. And so that came up with, these were the top cell-cell interactions that were predicted by that. And what I've done in red is highlighted proteins that have been identified in previous preeclampsia studies, bulk sequencing studies of decidua and preeclampsia as being associated with disease. And so it's kind of hard to quantify to what degree, but we saw there was a pretty striking enrichment in sort of preeclampsia associated genes that are associated with spiral artery remodeling, which probably isn't terribly surprising. So the last thing I'll show you real quick is just some of the stuff we've been doing with the glycan work. And so essentially, it's the same thing here. Aberrant glycosylation is a big driver of local invasion in cancer. And what we thought we would go and see is if some of these same glycans seem to be associated with trophoblast invasion into the decidua. So we had our MIBI images. We went and we did multi-glycan imaging with this new Bruker instrument that we've gotten. And we co-registered those and correlated those two data sets. What that allowed us to do was to see which glycans were correlated with trophoblast invasion. We saw a really tight correlation with highly branched glycans that look a lot like this one here. And so then, it's kind of to show you what we think is the vision for sort of making the use of all these techniques is if we have this image, from the spatial proteomic data, we have the transcriptomic profiles, and then that allows us to identify genes that are differentially expressed. But then if we compare also with the MALDI data, we're able to see which glycans are specifically upregulated by the EBTs. And then if we put all that together, we can start to correlate and find out which glycosyl transferases are associated with the upregulation of the glycans. And so we did that here. We found that just based on prior literature, we would expect to see this enzyme go up called MGAT5. And we saw a very stepwise increase in MGAT5 in the transcriptomic data from villus to attaching to interstitial trophoblast. And we're like, oh, this is really cool. But it turns out, someone had discovered this in the context of tumor models, like in 2000. So if you knock out MGAT5, you get much less local invasion in breast cancer models. And so with that, I'll stop. These are my usual long-winded explanations, but for the sake of time, I'll spare you those. Have you take any questions?
So, Michael, really excited to see the glycan work. So obviously I noticed a lot of sialic acid.
A lot of them, yeah.
Glycans. So I'll be very interested to.
We're about to run the preeclampsia TMA.
For the transcriptome, do you pick up Siglec-6 by chance? And
Yes.
was that differentially regulated
preterm.
over your time frame?
So in this time frame for Siglec-6, going from in the normal pregnancy samples that were in this study, I'll have to go look. I'm
Let's
not sure. I know in the comparison in the stuff in the TMA you helped us put together, we're comparing the preterm labor and preterm preeclampsia. They're definitely seeing Siglec-6 differentially expressed, specifically in attaching anchors. Because that's all the data is in that data set's just looking at attaching anchors. And so, but yeah, we definitely should. And I think it's going to be pretty exciting to correlate with these data, with the glycan data. It's going to be a lot of fun.
Yeah. I also noticed leptin was one of your.
Yeah, it was.
Good. Maybe you can help me figure my questions out.
Yeah.
Fascinating. And back?
Yeah, so the trophoblasts have a very different relationship with the arteries and the veins. So I'm wondering if you had any observations related to the veins.
Kind of. So there's some papers that seem to say that they're more invasive into sort of vascular lymphatic structures than we might think. So, you can find it. We've seen it too, but there's papers out there showing that you can find them invading within the vesicular glands. They can, and most people think they probably do invade the veins. The trick is, and the reason why we didn't try to make that distinction here is we didn't really feel confident in our ability to know for sure what was a remodeled artery versus just a vein. So we kind of went on the assumption that most of these were arteries, but there were some in there that definitely looked like they were probably venous system. And so we're thinking maybe go back and do this again with more specific endothelial markers. It allows it to differentiate between those better. But it's a good question about the preference for the vasculature. I mean, there's some papers though that seem to suggest that the arteries are actually the last to get invaded, and that they invade these other vascular tree structures first. And that would kind of make sense because you got to get rid of all the smooth muscle first, and that seems to be a gating event to let them in. But yeah.
So I'll end the discussion by just asking what I think is a crazy question, which is to say, given all the crossover and mechanism between cancer and placentation, is there any potential role for immunotherapy that's been developed for cancers and placental abnormalities?
I absolutely think so. I think that my guess, I have a very, very strong suspicion that if we had a magic box where we could look at women that have had, recurrent spontaneous abortion, where we know all the plumbing's fine, right? Plate and fallopian tubes, all that's okay. Ovulating, I would be willing to bet that there is an impairment to recruit these tolerance pathways, and if that's the case, I think that's a good thing, because I think that we're learning a lot about sort of adjuvant means to where I'd be pretty optimistic that we could, if we could figure out what that was, we could induce it, so.
Fascinating.
Yep.
Give Michael another hand. Thank you. So we are continuing to fall behind slightly, although Michael, you did a very good job at trying to keep us on schedule. So I'll make the next introduction as brief as possible. Jessica Ansari is a clinical assistant professor of anesthesiology, perioperative, and pain medicine, with a clinical focus on obstetrics, anesthesia, high pregnancies, and postpartum hemorrhage. And she's going to talk to us. Oh, I have to add one thing, which is that she did her medical education at UCSF. Seems to be a common theme here, since I was at UCSF myself for 30 years. And everybody else has that. And she's going to talk to us today about unlocking uterine atony. Just 1 minute for technical refinements.
All right. This happened to me last time.
Are you going to want to wander around, Jessica?
No, but I might have to just use the backup option of the slide somewhere else if we can't sort it out pretty quickly. But just because there's something strange. Unless anybody has good insights. Oh, you know what? Okay, perfect. If this doesn't work, I'm going to just see if I can put in right now. Stop sharing. All right, well, we'll just do it from yours. I don't want you guys to have to see all my scripting that I did earlier. That's too awkward.
Okay, I'll put my hand up when you've got
5
you
minutes.
started your discussion.
Amazing. So you guys can see this, correct? And you can hear me okay? Amazing. So I'm Jessie Ansari. As you said, I'm an obstetric anesthesiologist here at Stanford. And the title of my presentation is Unlocking Uterine Atony Through Calcium. That's the piece that isn't in the title on here. From bedside to bench and back again. And I'm thrilled to have this opportunity. So thanks for having me. Although I have a little imposter syndrome because I'm not a basic scientist. I have no disclosures.
You're becoming one.
I'm working on it. I'm going to get there. I'm going to be as impressive as y'all someday. Just watch. So as a framework of what I'll talk about, I'll briefly touch on what the clinical problem is in uterine atony. Then what was known about calcium and uterine contractility. Then I'm going to share with you guys some clinical trial results. And Dr. Yarmosh, my former fellow, is sitting in the audience who did this clinical trial with me. That we have results that are 2 weeks old and super exciting. So you're going to be the first to know. And I worked this into the presentation because it's brand new and not yet published. And then I'll launch into the translational work that I'm starting to do in collaboration with some brilliant folks from the Cornfield and Alvear labs. So very briefly, postpartum hemorrhage is a huge global public health problem. It's the leading global cause of maternal morbidity and mortality. And most recent estimates are that a person dies of postpartum hemorrhage in the world every 4 minutes. And uterine atony is thought to cause 70 to 80% of those cases of postpartum hemorrhage. So what is uterine atony? Very simply put, it's failure of the uterine smooth muscle to contract down firmly after the fetus and the placenta are delivered. And why does that matter? So as brilliant folks before me have pointed out, the placental bed has a robust blood supply, and the spiral arteries that feed that placental bed lack smooth muscle. So they do not contract down and stop bleeding when the placenta is delivered. The only thing that causes the bleeding to stop after a placenta is delivered is compression by the uterine smooth muscle, compressing that placental bed. So if the uterus doesn't contract normally, which it should do physiologically after delivery, you end up with continued bleeding into the cavity of the uterus. And another thing that's frustrating and that we know as clinicians is that up to 40% of cases of uterine atony are impossible to predict by clinical risk factors. And so it's not even like we can say, oh, these are all the high risk patients. Let's just make sure they deliver in amazing centers like Stanford, where we have good blood banks and specialists who can keep them alive, even if they have a profound atonic hemorrhage. This is something that can happen in any pregnancy to any pregnant patient. So what should happen physiologically? So after delivery, a flood of oxytocin is released from the posterior pituitary. It binds to the oxytocin receptor on uterine smooth muscle cells, leading to an increase in intracellular calcium and uterine smooth muscle contraction. And we as medical professionals augment this natural process by giving some synthetic oxytocin either intravenously or intramuscularly to help the process along after people deliver. If uterine atony occurs, as medical professionals, what we do is first we add more oxytocin. And when that doesn't work, we currently have 2 drugs that are FDA approved that we can use to try to fix the problem. These drugs are methyl-ergonovine and carbaprost. There are some problems with these drugs. So one problem is that they don't always work. In fact, a lot of the time they don't work. Another problem is that they are very costly and both require refrigeration, which severely limits their utility in lower resource and third world settings. The third is contraindications. So methyl-ergonovine is contraindicated in patients who have hypertension or preeclampsia. These are very common conditions in pregnancy. Carbaprost is contraindicated in patients who have asthma, another common condition. So a huge subset of our patients can't even receive these suboptimal drugs. And finally, they have horrific side effect profiles. Methyl-ergonovine causes nausea, vomiting, headache, and hypertension, which we can usually get patients through. Carbaprost causes profound nausea, vomiting, diarrhea, and sometimes bronchospasm. And the nausea, vomiting, and diarrhea occur in virtually 100% of recipients of this medication. So even if it works, it's an awful, miserable drug. So we desperately need new drugs. And this is my entire career goal, is to come up with those new drugs. As a side note, my obstetrician who delivered my 2 babies is sitting here, Dr. Coleman, and I jokingly, not jokingly, told her with my second and final baby that my only birth plan was hysterectomy before hemabate. Hemabate is the trade name for carbaprost because the side effects are truly so miserable. Yep, that was my birth plan. Because she's sitting here, I had to add it. She knows it's true. She can vouch. So I like to call this the hammer approach, right? So before any translational collaboration, during my fellowship and after, we came up with this hypothesis that sounds far too simple, which is, what if calcium is our next uterotonic agent? Just calcium through the IV. And so this was built on some actual data, right? So for decades, we've known from in vitro studies that when oxytocin causes an influx of intracellular calcium and causes smooth muscle contraction, most of that calcium is coming from outside the cell. Most of it is not coming from the sarcoplasmic reticulum. And that mechanism has never been sorted out, but it's very consistent across all different assays and studies. The other thing that we know that's a little more recent is that when we take human uterine tissue strips and we do myography with those strips, if you raise the calcium in solution above the physiologic ionized calcium concentration, those strips contract more frequently and with higher amplitude in response to oxytocin. So these basic science studies made us think maybe more is better. There were also some human observational studies that strengthened this hypothesis. On the left is a 2004 study that showed, pretty simple, that serum calcium levels are higher in patients who are in spontaneous labor than matched controls who are coming in for a scheduled cesarean delivery and are not in labor, suggesting that an increase in serum calcium has something to do with onset of spontaneous uterine contractions. And another more recent study published in 2021 showed that lower serum ionized calcium was associated with a higher risk of profound atonic postpartum hemorrhage. So lower might be worse and higher might be better. And enter our clinical trial. This is a phase 3 clinical trial that we just completed. And it was not just built on nothing. It was built on a pharmacokinetic study in which we figured out a dose of calcium that was well tolerated by patients. and raised the ionized calcium level in their serum to a safe range that was well tolerated, but was significantly higher than baseline. And then a pilot study to test our protocol and to generate a sample size for the study. So in this study, we were particularly studying patients who labored and received an oxytocin infusion in labor, which is commonly used to induce or augment spontaneous labor, and who subsequently required a cesarean delivery. The reason we chose this particular patient population is we know these patients are at the highest risk of atony. So about 50% of laboring patients who were receiving oxytocin and then have to go in for a cesarean delivery are going to have some degree of uterine atony and a postpartum hemorrhage with blood loss of over 1 liter. And so this is a great population. Our incredible clinical research coordinator, Mina Michelle, is sitting right back here in the audience, and she had to consent nearly 900 laboring patients to get to 120 in our study. And that's because most laboring patients don't need a C-section. So this phenom over here is amazing and made this possible. Patients were randomly assigned to receive 1 gram of dilute calcium chloride by a 10-minute infusion right after fetal delivery, or a saline placebo control. The clinical anesthesia and obstetric and nursing teams and the patient were blinded to the assignment. And the primary outcome was how much patients bled during C-section. So here are the results. Nobody's seen these. Please don't publish them. I really want to. Okay, so this is a violin plot of the distribution of our data, and this is in all comers. This is looking at the primary outcome, which was blood loss. And so what you can see is the red lines are medians, the blue lines are means, and there was an average effect size of a 250 ml reduction in the blood loss. We did not expect to see anything significant in this analysis. We actually powered this trial to exclude patients who bled for surgical reasons, like the incision in the uterus extending out to the sides toward arteries, because no drug can fix that. So we powered it for this subgroup, which is even more dramatic. So we took out the cases where there were surgical or other reasons that no drug could possibly fix that caused a contribution to the blood loss. Those were evenly distributed between groups, as you would expect in a good trial. And the effect size difference was massive. So we end up with a 360 ml difference in average blood loss, and a very significant, even lower bound of that 95% confidence interval. So super, super exciting. We can conclude from this small trial that if borne out in larger multi-center trials, which you better believe are next, a single dose of calcium chloride decreases blood loss from during intrapartum cesarean delivery. And this is exciting because a vial of calcium chloride costs $2.64, even at Stanford, where we mark everything up. It is shelf stable. It is readily available. It is FDA approved and used every day by anesthesiologists in the OR, so it is familiar. It's present on obstetric units for reversing magnesium toxicity. So very, very, very exciting. But our work isn't done, and nor is my talk. Did you have a question?
Just wanted to point out that the number of patients who had, near fatal amounts of
But I will say we didn't have very many of those. So this is why, of course, we have to do the larger multi-center. But thank you. I'll take it. I'll take it. I hope it plays out exactly like this in the larger.
Actually, another quick question.
Yes.
Do you have what the? Because I'm curious if the woman's calcium levels going in.
So in our pharmacokinetic and pilot study, we drew samples. To get this study done, especially since these types of cesareans are often done outside of normal operating hours, we didn't draw labs, but it was remarkably consistent. Like ionized calcium was almost identical across the board in our pilot study baseline. But our work isn't done. One, we have no idea why this works beyond the hammer, like, oh, maybe more calcium will work, which seems like almost idiotic in its simplicity, but wonderful if it does. So we don't know if this is an oxytocin dependent or independent mechanism. We don't know what types of channels this calcium is coming through. Is it L-type voltage gated channels that we try to hit with tocolytic calcium channel blockers, other types of channels? And even in our calcium group, we didn't completely prevent cases of uterine atony or postpartum hemorrhage. So we still need more studies. We need mechanistic studies, and we need to find more uterotonic drugs. And this is where translational research and basic science research is absolutely essential. And so recognizing this, almost 2 years ago now, I went on to, this is a very Stanford story, went on to NIH Reporter to find an expert in uterine contractility. And I found that 100 yards away in the BMI lab was the Cornfield and Elvira labs. And these guys are global experts in smooth muscle biology, calcium-mediated contractility, and funded with an R01 for uterine contractility. They happen to focus on the other end of the spectrum, which is preterm labor and trying to halt uterine contractility in the case of preterm labor. And so a really beautiful collaboration was born, and we've started centering a lot of our work on human myometrial tissue samples to try to get at some of these problems and questions. And so these are some of the questions we're trying to answer. But I'm going to keep moving since we're behind. We gather human uterine tissue samples from the hysterotomy site, so the superior midline edge of the incision made in the uterus during a C-section after the baby and placenta are delivered. We wash them in PBS, put them in media, and whisk them down the road to the Cornfield lab, where we sort to primary smooth muscle cell culture and also use the whole tissue samples for things like sequencing, transcriptomic analysis. A lot of our experiments have centered on calcium imaging using a Furo2 dye. And so these are some of our primary human smooth muscle cells. And the color change from blue to yellow or red indicates kind of an increase in intracellular calcium. You'll see this in graphs in the next few. So first, with our cells, with our primary uterine smooth muscle cells from human, we just wanted to confirm what has been shown by others, which is one, oxytocin causes an influx in intracellular calcium, and two, that is very attenuated if the extracellular calcium is low, and that's true here. Then we started doing some kind of unique and exciting work. Usually, most experiments that are published, at least in human smooth muscle cells, are from convenient samples that are from healthy, normal patients. In this experiment, we compared uterine tissue samples from patients who had normal contractility during their C-section and didn't bleed too much to patients who had profound uterine atony. And what we showed is that the oxytocin response in the smooth muscle cells from those patients was markedly diminished. And so that is what you'd expect, but it was awesome to see it in real life. And similarly, during C-sections, we ask our obstetricians periodically to put their hand on the top of the uterus and grade how well this uterus is contracting so we can decide if more oxytocin or uterotonics are needed on a scale from 0 to 10, a subjective scale. 0 is terrible, 10 is contracting great, we're in great shape. And what we showed was that the obstetrician's real-time in vivo assessment of how well a uterus contracts correlates pretty strongly with the dynamic calcium imaging response to oxytocin in our in vitro assay, which we couldn't believe, and really in some ways validates both assays, both the grading of the tone by the obstetricians and the dynamic calcium imaging and the rise in the intracellular calcium in response to oxytocin as good tools to use to try to assess how well the uterus smooth muscle is contracting. This is also something we're going to now have to try to restore with our new therapeutic kind of targets to try to restore a normal oxytocin response in cells from patients specifically with uterine atony. Other work we've done has centered on TRPV4 receptors. So Dr. Cornfield and Elvira's labs have done a lot of their pioneering work on this TRPV4 calcium permeable membrane receptor. And what they published and showed in the past is that in mice, TRPV4 increases in both expression and membrane localization through gestation, and that agonists cause contractions in mice, and that antagonists can delay labor. And we hypothesized that we might see the same effects in humans, and that agonists might be useful for atony, whereas antagonists might be useful for preterm labor. What we showed in our dynamic calcium imaging in our human samples is that even in the absence of oxytocin, TRPV4 causes a rise in intracellular calcium. And similarly, in a contractile assay in which these smooth muscle cells are layered on a collagen gel, TRPV4 agonists cause these cells to contract even without oxytocin. But interestingly and excitingly, TRPV4 antagonism, so blocking that TRPV4 channel, blocks the effects of oxytocin in smooth muscle cells. So it looks like potentially part of the mechanism by which oxytocin causes intracellular calcium to rise may be via TRPV4 channels. And this same same result bore out in our contractile assay. And then the last arm of our work has really focused on transcriptomic analysis, separating out patients who have profound uterine atony and the best matched controls by things like race, gestational age, body mass index, and comorbidities, and looking to find transcriptomic signals and differential gene expression in the patients who had atony that surprised us. And so far, we've just done preliminary experiments with bulk sequencing with 5 patients with very profound atony, 2 of whom required hysterectomies because their uterus just would not contract no matter what we did, and then the 5 best matched controls for them. And we found differential expression of many of the calcium signaling related genes. And what we're planning to do next is we're doing single nuclear RNA sequencing. Human smooth muscle cells are so large that single cell RNA sequencing doesn't work well. They're too big for the standard plates, but single nuclear RNA sequencing does work well. And so that is underway so we can deconvolute this data and also figure out which smooth muscle cell subtypes are most important for this response and for the development of uterine atony. So in summary, I covered a lot of stuff and some of it was not very basic science, but super exciting and I wanted to share it. So uterine atony is a big problem. I shared our clinical trial results that we're very excited about, suggesting that just giving calcium may help treat uterine atony, and then showed some of the work that I'm starting to do in collaboration with these brilliant folks in the Cornfield and Alvira labs. My goal is to go backwards in time and get a K award next and end up being almost as brilliant as some of y'all. I've got to acknowledge on the top row, the Cornfield and Alvira lab members. So Dr. Cornfield, his brilliant postdoc, Dr. Fornes, his amazing senior project scientist, Li Hua Ying, and Christina Alvira. I've got to acknowledge Dr. Yarmosh, who's over here, who was my fellow, who made this clinical trial possible. Dr. Obio is a former fellow who helped collect all these uterine tissue samples. Mina Michelle, my research coordinator, and a current fellow who's carrying this work forward, Dr. Lydia Esther Martinez. And then my funding sources, my division chief, and my supportive chair. And sorry for delaying things with technical issues. Terrible.
Thank you, Jessica, for a fascinating story.
Yeah.
I just wanted to ask you, does the fact that many of your atony patients come out of oxytocin stimulated cohorts, is there any reason to think that that is using up the calcium or desensitizing them to calcium?
It may be, and certainly one of our other fields of next study is going to be calcium as a treatment rather than a prophylactic therapy in patients at risk. So we know that a lot of patients have no risk factors, right? And so it'd be really interesting to see if that bears out. All of our basic science work and our omics work is in non-laboring patients so far who had no risk factors, and so that's not playing a role there.
Fascinating. Other questions? Yes, Michael.
Hey, God, I gotta start. Hello. I love the talk. The question I had was, so the defect that it looks like you guys can do when you stimulate this calcium channel, when you supplement with calcium in the bioassays from the patients that had uterine atony, you have, even in an excess of calcium, you're seeing attenuated response. Is that correct?
Didn't do that yet.
Okay.
So that is a very obvious next step, is to see if we just increase the calcium in the bioassays. So we kind of worked, this was before we knew the results of the clinical trial, that we did that first assay just to see, is this matching with what we thought we knew, that extracellular calcium is necessary for oxytocin mediated kind of contraction.
Well, let's assume it doesn't fix it. I bet it won't. So here's my, I have two like off the wall questions. One is, is anything known with women that have uterine atony? Do they have trouble breastfeeding?
You know, that's a good question for a maternal-fetal medicine expert. I know that patients who suffer hemorrhage and patients who have a cesarean do have usually a delayed onset of like their milk coming in, but I don't know more detail than that.
So in an elective section, the milk is delayed in terms of coming in. In terms of somebody who's had severe anemia, we also think that impacts production. But I always thought of that more as fluid. balance, but maybe
Oh, I was just,
maybe
the
that's
reason I
something.
was
You're
asking.
thinking of smooth muscle cell from.
Yeah, well, I was just thinking, like, one, is this receptor, is it uterine specific or not?
TRPV4 is found everywhere.
Oh, it is.
It is everywhere.
Oh,
yeah.
okay.
So like
Okay.
there are clinical trials out there using TRPV4 antagonists to treat heart failure.
Oh, interesting.
They're
Okay.
they're really well characterized.
So you don't think it's mediating like an oxytocin specific? It's an oxytocin specific calcium
It's
channel.
not even calcium specific. It's like a non-specific ion channel.
Okay.
It's implicated in chronic pain processes and neurodevelopment. It's kind of all over the place.
So if you use target the TRPV4 channel, are you going to end up with your diarrhea again?
Maybe. Right? That's an important question, right? I hope not. But I would actually take
Hysterectomy.
diarrhea over death for a lot of questions. But yeah, no, it's a good question, right? And nothing's been FDA approved since carboprost in 1979. Nothing for uterine atony. 1979. We can do better. This kills people.
If there are no other questions, I would thank Jessica once again for an amazing talk. So we are admittedly running behind. We should be in our final question and answer period here, but it's been so fascinating to hear everybody's presentations that I think we can't penalize our last speaker. We're very much looking forward to hearing Kristy Redhorse talk. She's an associate professor in Stanford's Department of Biology and in the Institute for Stem Cell Biology and Regenerative Medicine. Dr. Redhorse is very distinguished as a Stanford faculty member for her work in coronary artery development, and very much looking forward to hearing your talk today.
Thank you. Okay, well, it's great to be here today. This has been some exciting talks. I did my PhD on spiral artery invasion, and this is really making me want to get back to it. And what I'm going to talk about today, is related to heart development. It's not in the maternal-fetal interface, but it's a pathway, as you'll see, CXCL12 and CXCR4 that I'm going to talk about that is related. Okay, so in my lab, we study how the coronary blood vessels of the heart form. And we're interested in all life stages of the heart and how the vasculature. We're interested in the very first sprouts that grow onto the heart muscle before it has its own vasculature, how these sprouts then mature into a artery, capillary, and vein network, and then how this network expands after birth when the heart expands 10 times in size from birth to adulthood in mice. We're interested in how the vessels respond to injury, and we have this pipeline in our lab in that we look for the developmental genes and try to re-express those in the adult to regenerate the arteries in disease settings. Today, I'm going to tell you a developmental story by a postdoc in my lab, Gaetano D'Amato, and he worked with Alessandra Vigilante at UCL College London and a MD PhD student who recently graduated, Ragini Finsalkar. So this story is going to involve the very first steps of sprouting that I referred to. And those first steps come from two different cell types. So the coronary vasculature, the arteries, capillary, and veins, come from primarily two different progenitor sources during development. The sinus venosus, which is a vein on the backside of the developing heart, and the endocardium, the cells that line the inside of the heart. And they migrate towards each other from outside in and inside out respectively and form the vasculature of the heart. And over time, over the last 10 years that I've had my lab, we've been interested in why have two progenitors. We've provided evidence that this allows you to have rapid myocardial growth during this mid-gestation period. That when you have a mutation in one of them, the other can compensate, so robustness to development. We showed that even though they're coming from two different places, transcriptionally at least, they converge, and also they're very similar to human heart development. But today I want to tell you about our questions about when do these progenitors emerge. And so the main question here is whether there is a continuous or restricted conversion from these progenitors to the coronary vessels, because as I mentioned, there's a huge expansion in the heart muscle and you have to expand this vascular network. So the first model that we considered was a continuous, where you can think of this heart development scheme and the fact that maybe these progenitor sources are constantly feeding in new cells as the heart grows. And this would be new vessels during heart growth are fed by constant progenitor contribution. And there was some evidence in the literature of this happening when we started this study, although there were some other interpretations of the data. The other model that we considered was a restricted model where perhaps there's just this one time point in development that you get progenitor conversion, and then the vessels that arise from that are what are expanding over time. And so the experiment that my postdoc at the time, Gaetano, designed here was to do a pulse chase lineage tracing. And so he lineage traced the progenitor. And in this case, we used the endocardium because that is the tools that we had at the time. And so here he's inducing the labeling at different time points in development of the endocardium, and then asking later on, when do you see then coronary blood vessels labeled? And so to be more specific technically, we used a BMX Cree-ER cell line with a Rosa Cree reporter and injected at different time points. And if we looked at the front side of the heart, and you can see down here, the white is marking the lineage tracing. Up top in the colors are when we pulsed the labeling event. And I hope you can appreciate that only at the very earlier pulsing dates did we get a lot of coronary vessel lineage tracing in this area. So the conclusion from this and many experiments coming afterwards are that the endocardium to coronary differentiation occurs in the Cannon model, early conversion, and largely before sprouting begins. Okay, now I'm going to skip this because we're running late, but it further showed more evidence that there is this model where it is really an early differentiation and then growth from that. And at the same time, this was also shown by two other labs, Ben Zhao and Carrie Alatalo's lab. Okay, so this was good information for us because, as I mentioned, we just like to know everything about coronary development in the heart, every detail. And so previous to this work in the lab, we'd shown that the sinus venosus pathway, this sprouting pathway, is driven by VEGF-C and Elabella. Elabella binds the apelin receptor, but that these pathways are not driving endocardium angiogenesis. And so we wondered why it's different, basically, and more specifically, what is the signal to cause these endocardial cells to sprout into the cardiac muscle? And so we had done a previous study where the one that we showed that Elabella was an important driver of SV sprouting. And we had hypothesized that this chemokine, CXCL12, is one that is going to drive angiogenesis from the endocardium. We provided evidence that for this model where the sinus venosus sprouting from the first is developmentally timed, so it doesn't matter what the environment is, you always turn on these genes. But the genes that are important for the endocardial sprouting, we hypothesized were physiologically responsive because those are that we saw to be important, and other labs have shown that VEGF-A was important for this pathway. Those are hypoxia inducible. And so one of the ones that we are hypothesizing here is CXCL12 signaling to CXCR4 was because we see it expressed in the right region. And the cool thing here is that Michael has left, but in the preprint that he showed, the very last figure is that the spiral artery connections are potentially between the trophoblasts expressing CXCL12 and the spiral arteries expressing CXCR4, and that this is a gene that seems to be up in preeclampsia. So that would be cool if we can use the tools that I'm going to talk about next to maybe focus a little on the placenta. But nonetheless, let me digress back to the heart, where we wanted to test this hypothesis. Like I said, the CXCL12 is expressed at the right place in the right time. These are expression data that might be confusing if you're not used to looking at the heart, but I just want to say the bottom line is that they're at the right place at the right time to guide endocardial angiogenesis. And so we went on to do mouse genetics, knocking out in different cell types, the receptor and the ligand, to ask the question, is CXCL12 guiding endocardial angiogenesis? And here's the results of one of those analyses, where in this particular case, we're deleting CXCL12 from the myocardium. And we're staining for endothelial cells, coronary endothelial cells, and for arteries. And I hope that you can appreciate here that the green endothelial cells are the same in number, whether we knocked out CXCL12 or not. And so it doesn't look like endocardial sprouting is requiring CXCL12 in the myocardium. But as you can probably see, there's no pink in the bottom, which is the artery. And so what did seem to be affected is the arteries. And so here's a specific analysis of this where we're staining for the arteries and doing another type of genetic knockout. We do so many of them. I don't want to bore you with every single specific CXCR4 deleted cell type and CXCL12 deleted cell type. The bottom line is when you take out this pathway, you delete specifically the endocardial derived artery development or inhibit it significantly. Okay, so because no. Well, so actually there's two venosus derived things. It's the arteries and the plexus. And so the arteries from the venosus will start to creep in some of our samples. So the sinus venosus derived vessels will compensate a little bit on the artery side. And you can see that here. And this artery growing in, it doesn't normally come in from there. This is from the sinus venosus. Trying to compensate. So this made us think about all the times we've looked at expression patterns and had a hypothesis that didn't turn out to be true when we knocked them out. And so we thought, well, maybe there's a better way to predict which of our pathways might be functioning, and that is to try to analyze where receptors are being activated instead of just where they're expressed. And in this case, we would want to look at CXCR4 activation, the receptor for CXCL12. And so we got a little bit of inspiration from Richard Proya's lab, who made a activation reporter for the S1P1 receptor. And Tim Law did a lot of work with this mouse in the vasculature showing it worked well. So in collaboration with Kathleen Cairn at UNC Chapel Hill, we made this CXCR4 tango mouse. So in this scenario, we have genetically engineered the CXCR4 gene to have this extra tail here, which has a cleavage site in the TTA transcriptional activator. And when you put this in combination with a mouse that has the activator sequence on the DNA driving expression of a reporter such as H2B-GFP, then when the ligand is around, then you will get GFP expression, and that's reporting activation. There's one more key component of this engineered system, and that is you also have to engineer a beta-arrestin attached to TEV protease, so that when the ligand binds, beta-arrestin comes in, it cleaves off this transcriptional activator. So we used this we looked at this during mouse development in the heart, and we found indeed CXCR expression does not mean activation. This is a section through a heart, but you can see here that there's expression of CXCR4 and only a subset of those cells are activating or are showing activation of this tango system. And when we quantify that, it's mainly the artery endothelial cells, consistent with the mouse knockout studies. So we think this is a little bit better or improved way to analyze where this pathway is being activated. And so now we made this mouse so that we could look at regeneration, because we showed that CXCL12 can induce artery development in an injury setting. But now I would really love to look at this reporter in the placenta, of course. Okay, so a question for us now was, does CXCR4 activation, since it's in arteries, what does it do there? Does it cause those endothelial cells to take on an artery fate? Is there any influence on transcription? And so we did single cell RNA sequencing of these CXCR4 knockouts. They have CXCR4 deleted from the endocardium and all of their progeny. And we found that, surprisingly, artery specification is not affected, which could make sense because it's a G protein coupled receptor important for migration. But it's nice to see in the data because we wondered this for a really long time. And it's not the cell state of being an artery cell. It seems to be incorporating and undergoing morphogenesis to make the actual arterial vessel. And that might be why the only transcriptional differences we saw were in large artery markers. So they don't make the large arteries. So they're probably not getting those signals to express those markers. Okay, so that was what we learned about CXCR4 signaling, but of course we are wondering, well, what is driving endocardial sprouting? And so Gaetano decided that he was going to take this feature that there is sprouting early, but not later, or there was conversion from the progenitor to the coronary vessels later, but not earlier, to try to figure out what might be some drivers. And so we had single cell RNA sequencing from these hearts that had been lineage traced. And we took a look at those, and consistent with the lineage tracing experiments that I showed you, we have the lineage tracing, and we have all the endothelial cells from these hearts at these different stages. And consistent with our lineage analysis, in this very early time point, we could see cells that, 5 minutes. We could see cells that were transcriptionally bridging the endocardium and the coronary vessels. So that's what you would expect if these cells are in transit to differentiate. But we didn't see that at E17.5, so that was consistent. And we know that we label in this cell population, which is the endocardium, and we're tracing into the coronary vessels. And so we know this is the trajectory differentiation. So what is specific to this early transition? Well, Gaetano looked at a bunch of different things, and the one that caught his eye the most was BMP2 expression. And that's because, as you can see here, it's so specific for this endocardium to coronary differentiation. And this is a secreted factor, so it wasn't what we were kind of looking for, a receptor, because these are the cells that are actually doing the sprouting. But the expression pattern was so specific to him, he wanted to follow up on this. And so the model with BMP2, which is going to be secreted during this transition phase, is that it then signals back to the endocardium to, once sprouting occurs, and a small amount is then going to be what we would hypothesize to be a positive feedback loop. So if that were true, we need to show that BMP actually stimulates sprouting from the endocardium. And he did this with an in vitro assay. And what he first found was that when he lineage labels the endocardial cells of the very early heart and then puts them in explant, we know that these usually will sprout. But when we have the vehicle or the BMP2, there's no sprouting. We know that they do sprout because if you add VEGF-A, the little sprouts will migrate out. But when we add BMP plus VEGF-A, that really was potentiated. And so this is the quantification to show that BMP2 can't do it on its own, but it really potentiates VEGF induced sprouting. So we think BMP2 might cause them to be competent, like inducing a fate change, which is what BMP does earlier in heart development to the endocardium. And then that fate change or state change allows them to respond to the VEGF-A in this way. So the final thing I want to show you is that we injected this into an injury model to see if we could induce endocardial sprouting at a time later in which it doesn't normally happen. And so I'll show you the neonatal mouse where Gaetano performed an experimental myocardial infarction by tying off the coronary artery. He injected a virus expressing either GFP or BMP2, and he saw that this increased angiogenesis from the endocardium. And I'll just note that I'm not telling you here that the injecting BMP2 without the injury didn't cause anything, which goes along with the in vitro data that you need VEGF-A around, because there's not as much VEGF-A at this time point until you cause ischemia with the myocardial infarction. So to sum up here, we showed that endocardial cells which differentiate into a coronary plexus and then artery, capillary, and vein. This is something that happens, the first differentiation happens very early and is restricted in the adult stages. This first step is activated by VEGF and BMP2. And it is CXCL12 and CXCR4 that is inducing artery differentiation. But if we have injury in BMP2, we can cause this restricted to go to the accessible. And so finally, this is my lab. Gaetano is gone now. He's a scientist at Merck, but he's in the front there. Thank you.
Thank you, Kristy. Fascinating talk, which really pulls it all together, which of issues that you've been working on for years and years. So it's really fascinating to see. I'm going to ask if there are questions here or from Zoom. So yeah, so having ruled out the CXCR system for as the trigger for where do you go next? Where do you?
Well, since we're kind of excited about this BMP2 and its ability, so the next thing we're going to do is to inject that into the adult with injury, because what I showed you was the neonatal heart, which is already halfway able to do many things. It's able to regenerate. So we're going to go see if we can induce the angiogenesis in the adults by BMP2. So we're pretty excited about this pathway.
Yeah, so that is one of the amazing things about the mouse neonate, which is this ability to regenerate, isn't it?
Mm-hmm. Yep.
I actually had a question, or I guess it's really clarification. So if the progenitor is only early on,
Mm-hmm.
and so that would be the stage that you'd usually have the BMP2
Yes. Mm-hmm.
on, but doesn't get traced later in development. How is because it almost feels like, how would you expect the BMP2 to revert? You've got that reversion. Do you have a sense of why that's possible?
Well, we think that our hypothesis is that the BMP2 is not naturally around later on. And that might be part of why you're not getting a lot of angiogenesis from the endocardium later on. But when you reintroduce it, then you can.
So those early progenitor cells are still there. They're just nascent, and that you're reactivating them by introducing BMP2 back?
They're either
Or-
still there, or the endocardium, which is a specialized endothelial cell type, is kind of like other endothelial cells, and with the right signals can be reactivated, throughout
Yeah,
life.
got it.
That's more what I would think. It's not like a nested progenitor.
Yeah,
But I could be wrong there.
cool.
So I may have missed this, but where's the BMP2 coming from in the native situation?
It's coming from the early sprouts.
Not from another
It
cell
could
type.
be from another cell type, but in our single cell sequencing data set, it was not there. We didn't have those cells because we isolated the endothelial cells.
Right. One of the things that puzzled me, and this is really very much off the wall, but to throw it out there anyway, was the potential role of tissue restricted macrophages, microglia or equivalent, in the heart, because I know there's some suggestion that the capacity for regeneration in the neonatal mouse correlates with the presence of these before they're exhausted.
Right. Yeah, those tissue specific or tissue resident macrophages, Corey Levine's work has shown are really important in resolving inflammation even in the adult, and are kind of the good guy macrophages in that scenario.
So they even persist into the adult.
Yes.
That's okay. I wasn't aware of that. So the loss of regenerative capacity can't be attributed to the loss of those cells if they persist into the adult.
Right. It just, when you take them away in the adult, the fibrotic response gets much worse.
Right.
Yeah.
Right.
And there's another imbalance with the other type of macrophages that favor the fibrotic response. But it's pretty complicated, or it's multifactorial why the adult heart doesn't regenerate. The cardiomyocytes don't proliferate anymore. The vasculature doesn't remodel as regularly or as readily. And so there's a lot of different reasons. Primarily the cardiomyocytes not proliferating.
I see. Right.
And more fibrotic response, more robust fibrotic response.
So you still have a job to
Yeah.
figure out.
Yeah.
There's still some questions left.
There's still questions left, but now I want to go to the placenta all of a sudden.
Wait, can I ask a question related to the cardiomyocytes before we move on? Actually, so I still learn something new every time I see your talk. Doesn't no matter how many times I see it. I was struck by the early image you showed of CXCL12 expression, because it looks like it's in the left ventricle and the outflow tract,
Right.
right? So does that mean what does that mean for the endocardium and how it contributes to the right ventricle? Or is this axis, the signaling axis, specific to the left ventricle because those cardiomyocytes seem to be expressing?
Yeah, no, it moves over time.
Oh, okay.
So those are kind of a marker of where the hypoxia is.
I see.
What we found. And yes, it's always in the outflow tract, and it's very important in guiding the coronaries to attach to their blood source, which is making these connections with the aorta.
I see.
But the sprouting does happen more into the left from the endocardium. It
I
goes
see.
into the septum first,
Yeah.
and then from the septum, it goes forward,
I see.
mostly in the left, a little bit in the right.
But the right ventricle is later.
It's just a little bit less. I'd
Less.
say,
I see. So just not from. I see. Okay, great. Thank
but
you.
it does move over time.
If there are no more questions, please join me in giving Kristy another hand. All the afternoon's speakers, and I'm going to turn everything back over to Virginia to close the session.
All right. Well, I want to thank everybody for hanging in there. I know we went over time. Your reward for staying around here is there is drinks, both alcoholic and non-alcoholic, out on the patio. So we will enjoy this beautiful day. Hopefully continue questions and interaction. And thank you again for coming. And we'll see you at the next symposium, which will be on the final domain of the Dunlevie Center, looking at clinical trials. So Jess, maybe your talk being originally at the end is a nice leeway or a segue for the next symposium, and you'll probably be tapped again. All right, very good. Thank you, everyone. Do you know what the final count was on Zoom?
Oh, 9 people. 9 people.